So the talk is going to be not just Mycoplasma Houstonova. That'll be the focus of most of the talk, but I'd like to slip in a little bit about some of the other mycoplasmas that also affect pigs today. So from the diagnostic lab, what would we want to have in a test, just in a general want? We would like to have a test that identifies all hosts soon after they're infected, that's 100% sensitive and specific, that detects all field isolates, distinguishes vaccinates from infected animals, and involves simple, easy sample collection, and is cost effective. So it's a pretty short list. It's pretty easy to accommodate that. Not really, and especially for mycoplasma. And really, my PhD is in Mycoplasma Houstonova, and it's in immunobiology, and I wanted to study more on the immunology side, but my thesis ended up being almost exclusively on looking at mycoplasma diagnostics because there was a need to address some of these deficiencies or better understand the tests that we have. For Mycoplasma Houstonova, it's a diagnostic challenge sometimes. When you send lungs like the ones on the left to the diagnostic lab, we're pretty efficient at diagnosing Mycoplasma Houstonova out of that. The bigger question and the bigger challenge is what do we do to identify Mycoplasma Houstonova in pigs, in the first 3 months of life? Is it positive, is it negative? And to try to determine what that percentage, what prevalence that is. And that's really what we struggle with. That's the population that we struggle with. And I don't like to think that all of my years of my thesis and not being able to answer all those questions were for nothing or that it's all my fault. I think that there's some significant problems that can be attributed to the bug itself. And mycoplasma, just as a general rule, is very difficult to culture. It ranges from tricky, Mycoplasma Houstonova is pretty tricky to grow, to impossible, like Mycoplasma suis, which I'll talk about later here, is impossible to date. The genetics of mycoplasma, a few species have been sequenced, and that's a problem to make a lot of improvements going forward at a molecular level. Houstonova has been sequenced, although over half of the genome is consisting of genes that we have no idea what the function is. And our ability to do knockout experiments where we delete a gene or add a gene to try to assign function, we haven't been able to do that. So that makes it have limited molecular tools. And then on the host side, mycoplasmas affect generally extracellular places, mucosal areas, and they're very niche adapted. They often colonize hard to sample places, which we'll spend a lot of time talking about today. The normal host defenses are impaired, and there have been studies where pigs had their thymus removed, and those pigs had as many organisms as other pigs, but they had fewer lesions, and it kind of points that the pathology that we see is host-induced, and if we could find a way to pigs to just ignore the mycoplasmas being there, they'd probably be in a better position. And that there's a lot of factors at onset that determine how quickly the onset of disease is. And so what we'll have is that pigs will be infected, and there's a lot of factors that will contribute to how quickly we'll see that problem, and that can be things like stocking density, other co-infections, the strain, the virulence of the strain. So there's a lot of things that can affect whether pigs are infected and we ever find out, or it comes sooner or later. So the risk factors that we just talked about are the production system, all in and all out, has a lower risk than continuous flow. And parity segregation, I've actually had conversations with people who did parity segregation and had sows gilts that were kept out away from the sows in the early parities, and they got rid of mycoplasma sort of by accident. So I think that there's something to be said for the issue of gilts just kind of refueling the fire. Stocking density, replacement animals, co-infections, and the Mycoplasma strain does seem to have an effect. There's some studies that will show that different strains of Mycoplasma Houstonova have different ranges of virulence and different growth characteristics, and some grow quicker or slower and progress to disease at different rates. In the environment, ventilation, temperature, and proximity to positive herds and other stressors all have an effect. In the pigs, there's not just Mycoplasma Houstonova. We think of that as just mostly causing pneumonia. It's been isolated from the brain. It's been isolated from endocarditis cases, but mostly we think of it as causing pneumonia. Mycoplasma hyarinus causes arthritis, polycerositis, and otitis media in young pigs. Houstonova usually causes arthritis in older pigs. Flocculari is generally considered a non-pathogen, although really all mycoplasmas are pathogenic at some level because they lack all of the biosynthetic pathways to make anything for themselves. And so if they kind of have to get everything from their host, then they must have some mechanisms to kind of steal those. And it's very genetically similar to Mycoplasma Houstonova, and so it's the mycoplasma that we like to think of as having the highest potential for cross-reacting and causing false positives in some of our assays. Mycoplasma suis, some of you might be thinking, is that a new mycoplasma that I should be aware of? Well, sort of. It's really Eperythrozoan suis that some geneticists decided a few years ago to reclassify into the Mycoplasma species. And because now that it's a mycoplasma, it's sort of a point of pride that I need to bring it under my research. So we'll end the talk with a little bit of talk about Mycoplasma suis. Mycoplasma Houstonova is an extracellular pathogen. It lives in the airways of the trachea and the bronchi, the large airways on the ciliated epithelium. It's difficult for the immune system to see it there, and so I think that that's part of why we don't get really strong seroconversion early on that's detectable. The spread is primarily, we think that the most important route is sow to piglet, and then however many piglets get infected, they go after weaning, and then there's piglet to piglet transfer, and then also the potential for airborne transmission exists. And Scott D has done some of that work in his research projects where has a source herd, and then they 9 kilometers away have found viable organism that was just airborne transmission. And the carrier state is something I think that we don't fully understand how long that is. There's something probably to this 9-month time frame that's used in a lot of European eradication models, and that there's been some work showing that mycoplasma is detectable in about that time frame. What I think we don't understand is what the bell-shaped curve looks like of pigs that resolve earlier, but how many pigs resolve it later than that. And if you stop at 9 months, how many pigs are in that population that are still carrying it? I think we don't understand that very well. So for Mycoplasma Houstonova, we like to do a lot of ELISAs. It's a really convenient, accessible assay on the inexpensive end of diagnostics for sure. And for Houstonova, there's essentially 2 different assays that we use in the U.S. There's the IDEX or Tween20, which is an indirect ELISA. It's a whole cell detergent extract, which is Tween20 is a detergent of whole culture. And so it's multiple antigens put onto a plate with not much selection. And then because of that, you'd think that there would be more of a potential for false positives. And that's where we start thinking of the potential for flocculari to be a problem, although that's been fairly difficult to show. The oxoid, which is formerly the DACO ELISA, is a competitive inhibition ELISA, and it's a single antigen. It's an internal protein, so don't really think it's correlated with protection. Those antibodies aren't really correlated with protection, but it's one protein. And so there's a potential for false negatives of pigs don't make antibodies to that one protein. And there are field cases where that seems to have been the case. And so for the most part, the IDEX and the Tween20 correlate, but there are cases where they don't correlate. And we have struggled to repeat those experimentally to try to sort those out. And we lack a really good confirmatory assay to date. As part of my thesis, we did a challenge study, which was 8 challenge groups of 6 pigs. 4 groups were challenged with 4 different strains of Mycoplasma Houstonova. 2 were infected with Mycoplasma flocculari, and 1 each were given hyarinus and Houstonova We collected serum and nasal swabs prior to challenge at 14, 28, 42, 56, 70, and 95 days post-challenge. And along the way, we necropsied 2 pigs from each of the groups at 28, 70, and 95 days post-challenge, and took nasal swabs, bronchial swabs, and alveolar lavage fluid. And so the first step in the analysis, we looked at the effective sample type and the site of collection on PCR results. And so we did Mycoplasma Houstonova PCR on nasal swab, bronchial swab, and bronchoalveolar lavage fluid over time. And for the nasal swab, the CT value, which a lower CT value indicates a stronger positive, it's sort of counterintuitive. But across the whole study, the nasal swabs had about the same CT value, whereas in the lung, the bronchial swab and alveolar lavage fluid were very similar to each other and were much stronger early on, decreasing over time. And I guess my take-home message from this is that I don't think quantitative PCR on nasal swabs is probably worth your money, because I don't think it's particularly reflective of what's going on in the lungs. So then we looked at serology from all of the pigs in this study, and we did the oxoid ELISA, the IDEX, and then also the tween 20 on all of the serum. And this is a summary of just all the pigs that were infected with the different strains of Houstonova, all compiled into one graph. And the bars on the bottom are the positive with the suspect shown additively above the bar. And what we saw was that early on, the oxoid detected positives 14 days post-challenge, and then was all positive at 28 days post-challenge and throughout. And early on, up to 28 days post-challenge, the indirect ELISAs weren't as sensitive to those serum antibody levels. And so in this time frame, this point of seroconversion, we have historically and consistently seen that the oxoid ELISA is more sensitive at that time point, but then after that, they're essentially equivalent. Another thing that we did to break down these results a little bit further were to look at the individual animals between 28 days and 95 days post-challenge, and looked at the IDEX and the tween 20, because we saw that they didn't always agree. And so there's a fairly high, almost 25%, a quarter of the animals that when tested by the 2 ELISAs, which should be essentially equivalent, they were just basically made with different strains of mycoplasma, that a quarter of the samples were positive for one, negative for the other, and there wasn't a real trend going one way or the other. And so I think that points to that the antigen in the plate makes a difference, and although there weren't any strains that weren't just flat out not detected, it seems a very pig-specific finding. And I think it's interesting to know that the false positive and false negative rate is probably something that we sort of underappreciate in those ELISAs. When we looked at so by the way, the flocculary, Hyarinus, and Houstonova groups, we found no cross-reaction in any of those groups, and so we're not able to substantiate any false positives due to any other species of mycoplasma. So this is the 4 strains of Mycoplasma Houstonova in the 4 different challenge groups, broken out as individual lines. And so this is to the IDEX ELISA, and the tween 20 was very similar. And the blue line is to one strain that seemed to have lower overall OD values compared to the other 3 strains. And so we had questions about whether that group was infected to the same extent. Maybe they had a lower infection, what we could do to sort that out. And so the blue group is this first group, correlates to the group that had the lower overall serum OD values. And this represents the percent lung pneumonia of the 2 pigs that were challenged at each time point. And at the 28 days post-challenge time point, which is the time that we expect to have the highest lung lesions based on our challenge model, the two blue pigs, the two pigs with that strain, had as high or higher total gross lesions as any of the other groups. And so we weren't really able to attribute that group just didn't get as affected. And so to further look at that, I made antigen based on that antigen to that strain, grew up and made plates, ELISA plates, so that it wouldn't be a heterologous comparison, it'd be homologous antibodies to that antigen strain. And so the top one is just the graph repeated of the tween 20 results using 232, which is our standard antigen. And then below is the same serum reacting against the antigen made by the blue strain, and we see the same trend. There's an improvement, but there's still the same trend. And so our conclusions were based actually on also Western blot analysis that the pigs just didn't seem to respond immunologically as much to that strain and didn't produce antibodies to that one strain. And so I don't know what implications that has to the field, but I think that we may also be underrepresenting serum antibody responses in the field to some isolates. And so for the summary for the ELISAs, they really are better on a herd basis. They are not very reliable on an individual pig basis, at least all the time. And there's no confirmatory test really to sort out when we do have that question. There's no correlation between the OD values and individual protection with current tests. So you can't look at the pigs that have the higher OD values and say that they're more protected. And that's because one ELISA is to an internal protein that's not correlated with protection, and the other ELISA is really to all the proteins. And if we knew which proteins were the really important protective proteins and we put only those into our ELISA, it would probably have a lot better correlation, but that's not currently what we have. And so the future of oral fluid ELISAs, I get that question periodically, because we're doing oral fluid ELISAs on PERS, and the work in Dr. Zimmerman's lab has shown that because there's a higher number of different isotypes in oral fluids, that we can detect antibodies to PERS sooner in oral fluids than we can in serum. And all I can say on the mycoplasma front is that we are able to detect antibodies in oral fluid, but we haven't been able to get over the leap of how you finally validate that assay, because we'll need to have specific oral fluids from pigs challenged only with hyorinus, only with flocculary, only with some of these other mycoplasmas to really improve that specificity, because I think because oral fluids have IgA and IgM and these other isotypes, we may be having potentially more false positives that we also won't be able to sort out. So we have our work cut out for us on that, but I think that that's something to look forward to, because potentially we'll be able to detect antibodies sooner with it. This is a study done by Marina Sabila in Spain. It was a chronological study of M. hyo infection in the field, and it included about 450 pigs. And they do a different ELISA in Spain than we do, but it's similar. And really I just wanted to highlight that in this study on a much larger, broader numbers, that the blue and the pink are vaccinated pigs. The green is negative pigs, so unvaccinated pigs, just naturally infected. And in that study, it took almost 12 weeks to be able to be detected by natural infection. And she also did nasal swabs. And so PCR nasal swabs also took 9 to 12 weeks to be detected by PCR. And so there's this window of 3 months where we really struggle to identify positive pigs, but we're using that time frame to make a lot of decisions on pigs, of selling them as negative herd stock and replacement animals. And so one of the things I tried to do was to see if I could optimize and maximize the tests that we have. And I had a summer student who kind of put her whole summer into trying to find out if she could really maximize our potential to do PCR and look at the potential for oral fluids for PCR to detect mycoplasma. And so we looked from start to finish, from sample collection to nucleic acid extraction in the lab, and looking at the PCR assay itself. And so really, it does matter the swab that you use for detection and collection. And there's sort of been, I've seen some studies where people have done cotton swabs and different kinds of swabs, and the kind of the worst swab, it's one of the most common swabs, is a bleached cotton swab on a wood applicator, and it's bad because the cotton has bleach and the wood applicator has formaldehyde. And there's a study, a study where someone just made a lawn of bacteria on a blood agar plate and just laid the swab on it, and there was this zone of inhibition. So I think it does matter for culture, it does matter for PCR, and it doesn't matter for everything. So if the sensitivity isn't going to be an issue, it might not matter what you use. But when it is an issue, I think one of the better swabs you can use, they're called flocked swabs. They are flocked like a Christmas tree, and their claim to fame is that they collect as much as other swab, but they release more into the media. So the commercial here is that they dipped it into blue dye and then dipped it into water, and you can see that the flock swab is darker blue. And then we also wanted to look at how good oral fluids compared to sort of the standard, which is nasal swabs in the field. And so just a small comparison that we had some pigs that were challenged with mycoplasma, and so at necropsy we stuck a cotton swab in one nostril and a flock swab in the other nostril, and then just did PCR, and found that the flock swab detected a few more, and that the CT values were considerably better for the flock swabs. And in PCR, 3 CTs is a 10-fold change, so 3 CT difference is 10 times better. And so, like I said, that doesn't matter for everything that and they're a little bit more expensive, but when sensitivity really matters, it's probably worth the effort. So we did a field trial, and we collected samples in conjunction with a vaccine trial in the field. There were 6 pens of 52 pigs that we used, and two pigs from each of the pens were challenged as cedar pigs. The herd was known to have some mycoplasma in it, but it was done to sort of have a more consistent challenge for the vaccine trial. So we did multiple collection points. We collected nasal swabs and oral fluids for 8 swabs per pen, so 48 total per time point. And oral fluids, we collected 1 per pen. And in that time, we didn't collect nasal swabs from any of the cedar pigs, and when oral fluids were being collected, we put the cedar pigs out into the alleyway, so they weren't part of the sample collection. And then we collected them at 6 points throughout the study, 4 weeks pre-challenge, and then up to 12 weeks post-challenge. And so the test results for the contact pigs were that the red bars are nasal swabs and the green bars are oral fluids. And so early on, we got nasal swabs positive, but we knew that the herd did have some positives in it. And then 4 weeks, we detected a few nasal swabs, and really didn't detect much until 8 to 12 weeks, which again, I think is a little bit disheartening because we'd like to be able to find them sooner than that. And nasal swabs and oral fluids were somewhat similar. The nasal swabs had about 75% at 8 weeks and 50% at 12 weeks, but the oral fluids were all positive. And for sure, it's a lot cheaper to do 1 PCR on 1 oral fluid than it is to do 48 PCRs. So as a cost comparison, it's a lot better maybe to do oral fluids. And as a further comparison, we looked at the CT values from the nasal swabs versus the oral fluids, and the CT values from the oral fluids were considerably lower, stronger positives than they were for nasal swabs. So I think that that's sort of a vote for oral fluids as a diagnostic tool. But I would like to have you consider another sample type. There was a study done in 2010 in France by Feblet, and they compared nasal swabs, oral pharyngeal brushing, tracheobronchial swabbing, and tracheobronchial washing. And they found that the tracheobronchial washing and the swabbing were considerably more sensitive than nasal swabs and oral pharyngeal brushing, which potentially could be thought of as similar to oral fluids, although it's not an entirely equivalent sample. But they found that the tracheobronchial swab was 3.5 times more sensitive than nasal swabs. And this graph is from 60 pigs sampled that were naturally exposed. And the 3 bars are really just 3 different statistical analyses to try to do this comparison as we try to put statistics to our studies. And so really it's the same 60 pigs represented 3 times. But the tracheobronchial swabbing, I know, is going to be something that's going to meet resistance in the field. And so I wanted to take a quote from her discussion, and she says, "As far as practical aspects are concerned, swabbing the tracheobronchial area with a sterile catheter is almost as convenient as obtaining a nasal swab under field conditions and only requires adding a gag to the sampling equipment." And I'd like to say that I wanted to make a video to show that I think it really isn't that hard to do and isn't as daunting. And if you, I mean, it has to be a pig that you have to care about, that you don't want to kill and would really want to know what its status is. But I think that a tracheobronchial swab is as good or maybe better than lung because the study by Kurth shows that a tracheobronchial swab and a BAL can be better than lung. So I guess it's something to think about, and if anybody wants to pursue this technique, I think I'm going to try to make a video and put it up somewhere, and I'd be happy to show anyone. It's similar to the method that we use to challenge pigs for Mycoplasma Houstonova, which is putting a feeding tube into the trachea. It's really, I think, an accessible diagnostic sample that maybe we should consider. Another thing to discuss is field isolate variability. And so I alluded to a little bit that there have been some studies done in my lab and in a couple other labs that show that different strains do seem to repeatedly have different levels of virulence. And I think notably, there's one study where there's sort of a low virulent strain and a high virulent strain, and they tried to vaccinate pigs by giving them the low virulent strain followed by the high virulent strain, and it actually didn't work very well. And another thing that they figured out was that the low virulent strain maybe wasn't as low virulent as they thought. It just took longer to become progress to a higher level of disease. And so I think that there's just some strains that don't behave the same as all the others and are low virulent, and what we do with that, I think, is yet to be sorted out. And there are some typing methods, and so we're really good at showing that the mycoplasmas are different from each other by using a lot of different assays. But what we have not been able to do is make the correlation of that it's predictive. So we show them that they're different, but we're not able to say that it's more virulent or that it's going to be a problem or that your vaccine isn't going to be protective, or we haven't been able to make any of that correlation except to say that they're different from each other. And so it's useful maybe in epidemiological studies, but we haven't figured out a way to kind of go past that. And most of the studies that were done in Switzerland, and it's basically eradicated there. And so when it shows up, it's generally from a source herd like a feral pig. And so it's one strain that blows up in an area. And there's some studies done in Germany that I don't think are published yet, that were presented this summer at a meeting, where they found multiple strains within one herd, and they also found multiple strains within one pig. So I think what we do with this typing going forward deserves consideration. And then I just want to do some comparison to what we do in sequencing for like influenza, which is 8 genes, 13,000 base pairs, and then codes for 11 proteins. And just to compare that to mycoplasma, which is, even though it's a really small organism, is 900,000 base pairs, and it codes for almost 700 proteins. And when we do these sequence typing, it's usually a small portion of a few genes or one gene, and so it's not really saying much about the rest of the organism. And so For influenza, if we're sequencing one of 2 genes that's on the outside and we struggle to know what to do with that information, it's compounded even more with what we do with it for mycoplasma. So really, we have some good diagnostics, but really what to do with them depends on what your question is. And if our ability to answer it depends on the bug and the host. And so if the question are things like, is mycoplasma causing disease in this herd? I think we're pretty efficient at answering that question. But questions like, how's my vaccine compliance? Is this herd negative? What are my replacement pigs negative? How can I confirm serology results? How can I confirm the status for export? And am I dealing with a new strain? Are some of the questions that we deal with. And for vaccine-related questions, trying to use serology for vaccine compliance, really, ELISAs are better on a herd-wide basis and aren't going to be very effective for individual pigs. And can I tell if pigs are protected? There's not really a good correlation in serology, so the answer for that's not really. My replacement pigs, are they negative? So there's this black box, this window of time where maternal antibodies wane and they're producing their own infection, their own immune response. And there's this time frame of several weeks where they're going to be ELISA negative, but probably at least some of them are going to be positive pigs. And so I think that it's time, or it's probably warranted to look to the sow. And that if we're not going to be able to have a very good feel for prevalence at weaning, which work by Fano shows that prevalence at weaning seems to correlate with disease levels at slaughter, but we're not very good at being able to detect organisms at that level, then we probably need to be looking up at the sow and maybe doing some swabbing of the sow and trying to get prevalence there. Or it's also somewhat effective to look at antibody levels in 1 to 2 week old pigs if you're not vaccinating in the sow herd, because all of the antibodies that those piglets have came from maternal antibodies. And so if they're very high, then that's an indication that the sow herd is probably unstable and maybe due for pulse medication or some other method of to decrease the organism level. And so that's what I do. If I'm going to be looking at a source herd for negative pigs, is that I like to look at the piglets in non-vaccinated systems. And you really can't do one point in time testing to say that a herd is negative. It really is multiple testing and gaining confidence, because serology isn't telling you about today, it's telling about a few weeks ago. And so it's gaining confidence over time. For PCR, I don't tend to have the same worry about potentials for false positives and what to do with them. For PCR, I like to say if it's positive, it's positive. If it's negative, that's a maybe, because we worry about the false negatives. And sample type is certainly important. When I get asked about pooling, and that's not just for mycoplasma, pooling is about risk and it's about prevalence. And if all of the pigs have the same level and you pool them, it doesn't matter. And if one positive in a group is going to be really strongly positive, then it doesn't matter. Like, we pool very high numbers for BVD PI calves, because really you could dump that blood into a pool and it would still be positive. So if one individual positive is very strong, you'll find it. It's when they're very weak or there's very few and you're going to miss them, and that has to do with risk. So how do I confirm negative status? Well, there's no confirmatory assays by single point in time. Use serology over time and PCR for low sensitivity in antemortem samples. Confirming Western blots, there's been one PCR or one Western blot that was described, but it's not offered anywhere, and it wasn't really validated. So really, we don't have a way to sort out Western blots except for PCR to identify the organism. And so that's when I break out the special swabs sometimes. By dealing with a new strain, so there's a lot of typing assays that have been described. They're useful in epidemiological investigations, but none are predictive of virulence or vaccine protection. And so to switch gears to the arthritis-inducing mycoplasmas, Mycoplasma hyarinus causes arthritis and polycerositis in young pigs, and we generally think of that up to 12 weeks of age. And Houstenoviae is arthritis in older pigs, more like 12 to 20 weeks of age, and we think that maternal antibody has something to do with protection early on, is some of that change in the time frame that we see disease. And it generally affects much bigger pigs, 100 to 150 pounds. So this is the incidence of Mycoplasma hyarinus and Houstenoviae at the ISU-VDL over time from about 2003 to present. And so the lighter bars are hyarinus and the older bars are Houstenoviae in weeks of age. And so just sort of demonstrating that in clinical cases we find hyarinus early on and Houstenoviae later on. And the overall incidence of arthritis cases at the ISU-VDL from 2003 to current is the total bar is the total number of cases with arthritis. The darker blue bar is Houstenoviae and the lighter blue bar is hyarinus. And we see that in the last few years there's been an increase in diagnosis at the ISU-VDL, but in reality that could be two things. It could be that truly that there is an increase of these two organisms in the field, but in 2008 we added PCR, so I think that's kind of a lot of it as we started looking for it a little more often. For diagnosis, we don't have serology for either one of those that we use anywhere except in research labs, and it doesn't correlate to disease because a lot of these two organisms are very ubiquitous, and so antibodies can be in pigs or not be in pigs, and they don't really correlate to disease. Culture would be the gold standard. Both of these are mycoplasmas that I prefer to culture over Houstenoviae, but they take up to 2 weeks and aren't always successful. The cultures have to be identified, and we do that by PCR, and it's not available everywhere, and it has to be specifically requested. So I've heard practitioners say that they're negative for mycoplasma because it never gets cultured, and you have to specifically request a specific culture, so you'll never get it by culture. And then PCR was added in 2008. So Mycoplasma Houstenoviae is something that I think that is on people's radar screen a little bit more in the last couple of years. The progression disease is age-related. It seems to be immunity-related, correlating with maternal antibody waning. Dick Ross did a lot of work on this. He discovered it in the 50s and did a lot of work that associated it with heavily muscled joints, and that there was some breed predilection, that the breeds that had poorer conformation or more heavily muscled seemed to have these problems more. And since it's such a ubiquitous organism, and you can find it in you can find it in joints of pigs that aren't affected, it seems to be that there's a trigger. And so it's kind of waiting for a stress event. I mean, nowadays when I get a call about it, I often hear that it's in my best group of gilts that are growing the fastest. And so my theory for that is that we're growing pigs faster than their joints can keep up, and that it's a predisposing factor to progression to disease, or that there's stress of moving or mixing. And then there does seem to be a correlation to virulence factors. Usual recovery is within 1 to 2 weeks, although some become chronically affected, and generally they have fundamental joint problems that, like OCD, that are hard to resolve. We treat it with antibiotics and anti-inflammatories. This was a study done in Copenhagen, and they just compared 3 herds, and I just wanted to show that there's different progressions in different herds that could all be positive. And so in herd 1, all the older pigs were infected, but it didn't seem to transfer to the younger piglets. In herd 2, all ages were carriers, and tonsil carriers is where you find it, but no disease or antibodies were detected in the grow-finished pigs, although they were all carriers. And in herd 3, all ages were carriers, and then they found disease in the grow-finish, and some seroconverted and some didn't. Some seroconverted before the disease and some converted after. So it's I think the diagnostics for it are a little bit confusing, and the ways to predict whether you're going to have a problem or not, I think, are also are difficult to sort out. And then I have a student, Kimberly Schultz, who's presenting on MIC testing that the ISU-BDL will be offering for Hyosinoviae, and so she'll be presenting this at the student seminar. So this brings me back to Mycoplasma suis. It's formerly known as eperithozoan suis, and the disease is still called eperithozoanosis, which is interesting. It's a bloodborne pathogen. In 2002, it was reclassified to the genus Mycoplasma, and it's an uncultivable organism, so it's only passaged in splenectomized pigs. Prior to the 1970s, it was diagnosed by basically herd history and clinical signs and blood smears, which are shown here. Acridine orange will diagnose it. Complement fixation test was really insensitive, and so for a long time, it was considered to be low morbidity and high mortality in feeder pigs. In the mid-1970s, we got an indirect hemagglutination assay, but it was kind of prone to false positives. And so then it was probably overdiagnosed, and we didn't know if it was high morbidity or low, and if the mortality was low because the mortality rate was affected by widespread use of arsenic acid, which essentially kept the disease under wraps until about 1986 when we quit using arsenic acid. Then there was a spike in interest in cases, but it was sort of forgotten because the diagnostics for it were kind of terrible. And it causes such a wide spectrum of disease, it's kind of hard to attribute positive with this cause and effect. But it does affect all ages of pigs. It causes a life-threatening hemolytic anemia and hypoglycemia. And the hypoglycemia is really unique to the pigs because lots of species get these hemoplasmas, but the hypoglycemia is sort of unique to pigs, and it causes acute death where they just go into hypoglycemic coma and die acutely for no apparent reason. It can also cause a mild chronic anemia in piglets, such as ill thrift, and feeder piglets usually just have immune suppression, retarded growth, seems to predispose to other diseases. In sows, infertility and decreased lactation have been described. And then in 2007, there was a PCR developed for this test, or for this bug, which got our interest. And there was also a assay, or a study done in Germany of slaughter pigs, and they found that like 40% of the herds that were surveyed were positive. And when they took out the pigs that were PCR positive and PCR negative and did some just basic hematology, such as hematocrit, hemoglobin, and erythrocyte counts and bilirubin, they found that although not statistically different, there was certainly a trend in PCR positive pigs to have lower blood values than PCR negative pigs, which you would expect, and these were herds that were considered normal and healthy. But I guess having a blood parasite is probably not good for you. And so what about some clinical cases? Since the ISU-BDL sort of has been offering this test, I thought that we would find this in clinical cases in these anemic pigs that are getting iron that we're not able to diagnose. And I've been surprised that the cases that I have worked up have been in sows failing to lactate, where the sow farrows normally and doesn't come into lactation for a week or so, but in that time the piglets starve. And so in those herds we've gone in with antibiotics prior to farrowing and have been able to get them over the hump, because antibiotics don't clear the organism, and you're probably going to find a high prevalence, but it'll only be a problem in certain stressful time points, such as farrowing. And if all it takes is feeding more consistently to keep the glucose level up and putting in some antibiotics, you probably could manage through it and maybe sort out some problems that were hidden, because to diagnose this, it really requires specifically looking for it.